Okay, now we're set to deal with the double crossover situation. So, as before with the single crossover situation, on this side of the synapsis we've got the little a that's recombined onto a translocated chromosome, and over here we have the little b which is recombined onto a translocated, and also the big B recombined onto a normal structure, and here we have a big A that's recombined onto a normal structure. Okay, now depending on the segregation patterns, we could potentially save any of those possible outputs, but we'll just do one example. Okay, so let's think about what's going on in meiosis 1. Let's start with this side of the equation here. So here we have a top chromosome which was not involved in the crossover event, and now down here we have that same little a allele, but now recombined onto a translocated structure. Okay. Down here in the other cell, we have the lower chromatid, which was not involved, which has a big A allele on a translocated structure, and also has the normal structure now with the big A allele on it as well. So that's what's going on the one side for the one chromosome pair. Now we need to decide what we're interested in segregating with these recombinants. So if we have, say, a little a which is now on a translocated structure, if we're interested in saving that one, we need to have something that now provides the translocated structure. So for example, with this crossover, we could have the little b which segregates with that. So if this is going up and we want to save something on that side, we need to segregate this one up as well. Okay, so that puts this chromosome into this cell, whereas the top has the translocated structure, so it's got the little b allele on it, and has the translocated structure. And then the bottom one is the little b allele on the non-translocated structure. So there's that one cell after meiosis 1. Okay, for this lower cell on this side, that means that we've taken this one along for the ride. So this one has the big B. So let's just do the top chromatid first. Has the big B on a translocated structure. And then on the one that wasn't involved, the one that was involved in the crossover, we have a big B now on a normal structure. And that's this cell after meiosis 1. So let's just trace that through, make sure we've done this correctly. So top chromatid, big B with the translocated structure, and then big B picking up a normal structure. Okay, and for this side, again, tracing it through, top chromatid not involved, lower chromatid, little a, and then the translocated structure. And then what we're segregating with that, top chromatid has little b and then the translocated structure, lower chromatid has the little b with the normal chromosome 2 structure. Okay, so we're okay for meiosis 1, and now it's time to take these on to meiosis 2. Now, the issue here in this case is because there are different possibilities now on both chromosome sets, we need to be careful about how we do the segregation for meiosis 2 as well. If we're interested in saving, say, the little a on the translocated structure, which is the result of one of these crossovers, so that's what we're interested in saving, then we need to make sure we pair the complementary structure with that. So in this case, if that one goes up here, then we need to segregate this one along with it. And that one can go down and go with that one as well. So if we carry this around and draw this out, then after meiosis 2, the top chromatid here is, or the chromatid here, sorry, that goes up, is this little a on a translocated structure, and at the same time we've got the little b on the translocated second chromosome structure. Okay, so there's that first cell after meiosis 2. The ones that go down is as follows. So we have the little a on a normal structure, and at the same time we have a little b on a normal structure as well. Okay, so that's going to be that outcome there. Okay, let's go on to this one here. Again, we're interested in saving the results of what's gone on based on the crossovers. So here we had the big A that crossed over onto a normal structure, so let's save that one. So if we take that one up and the other down, then in order to save it, we will pair that one with it. So again, whether or not something survives or not at this stage is dependent on segregation at meiosis II. It could well be that these combinations, these events, can get lost if the appropriate segregation pattern isn't followed. Typically in a question, what I'll ask you to do is to show a meiosis that produces a particular outcome, a certain type of progeny, and as a result you know that the segregation pattern had to work to allow that combination to survive. So you're only responsible for drawing the ones that actually show the event that we're interested in. Okay, for this lower set of cells, first off down here we have the big A on a normal structure, and we have segregating with it the big B on a normal structure. Okay, that's new. We haven't seen that before, and that's going to work. Likewise, we also haven't seen a little a and a little b on translocated structures before. Okay, so those are new as well. And then, last but not least, we have the translocated structure with the big A, and segregating with it is the translocated version of chromosome 2 carrying the big B. Okay, so little a and little b, that's genetically not recombinant because that was one of the inputs, but the input brought little a and little b on normal chromosome structures. And now here we have them as recombined onto complementary translocated structures. So that's the result of a double crossover event. Similarly, big A and big B together is not recombinant because that's the same as this input here genetically, but these alleles came in on translocated structures, and now a double crossover event has put them onto normal structures. Okay, let's summarize a bit, and I guess I can take these crossover events off. This reciprocal translocation heterozygote, as we mentioned before, is also a dihybrid, and like any dihybrid, it will produce the 4 possible outcomes. So here are the ones genetically, these are the non-recombinants, and these are the recombinants. Now, what makes this different, though, because it's a reciprocal translocation heterozygote, is that now each of these categories will be split into 2 different groups. There will be normal structures and also translocated chromosomes or translocated structures. And that will apply to every single one of these categories. So as a result, a reciprocal translocation heterozygote that is genetically dihybrid for loci that are involved with the reciprocal translocation, so these loci are on the chromosomes that have swapped tibs, you will now see 8 categories, just like what we saw with a trihybrid test cross. Remember how we talked about region 1 and region 2? The single crossover in region 1, single crossover in region 2, no crossovers, that sort of thing, just like what we saw with the 3-point test cross to give us the 8 categories. These same events now in these different regions will give us the 8 different categories here. So 4 genotype categories, but now each category either on normal structures or on translocated structures. And remember, when we take these gametes, say we're taking this organism and we're test crossing it to say little a, little b on normal structures, we can identify which categories have translocated structures because they themselves will be semi-sterile. And the ones on normal structures, because this gamete coming in is just a little a, little b gamete on normal structures, if we have normal structures from this gamete coming out of this, then this organism will have fully normal structures, will not be a translocation heterozygote, and as a result will be fully fertile. So with these 8 different categories, 4 of them will be fully fertile and 4 of them will be semi-sterile. And there will be a semi-sterile and fully fertile category for each genetic genotype category. And it's your responsibility to know how to decide if I'm, say, dealing with a semi-sterile offspring with this genotype, that means a big A and a big B on translocated structures that are now paired up with the normal structures that came from the tester. So you can treat it just like a 3-point test cross, or with region 1 and region 2, like we did when we were trying to go from a map to data. If you're given data, you can do this just like a 3-point test cross, except what you're doing is you're mapping the first locus to the breakpoint as region 1, the second locus to the breakpoint as region 2, and the double crossover events are simply producing the non-recombinant genotypes on the alternate structure. Okay, now that we've discussed deletion heterozygotes, inversion heterozygotes, and translocation heterozygotes, let's just summarize some of their salient features that will help you diagnose whether or not a question that you're dealing with involves one of these 3 options. First off, cytology at synapsis. If you have information that gives you a handle on what's going on here, a deletion heterozygote has a loop with only one chromosome that's looping and the other one's not part of the loop. Inversion heterozygotes have both homologs within the loop, all 4 chromatids, and translocation heterozygotes obviously have that cruciform structure that we've been talking about. Genetically, and this is something that's usually quite important for a genetics question, is the effects that these have genetically. For both inversion and deletion heterozygotes, we see a reduction in recombination for linked loci because we've removed some space that's available for single crossover events, either entirely or because we have sterility problems when there's a single crossover inside an inversion loop. With a translocation heterozygote, we see unlinked loci that suddenly appear as if they're linked with one another. And the reason for that is that we require a crossover now to provide alternate chromosome structures to get recombinant genotypes for the different loci that are on the different chromosomes. The sterility issue is also quite important for genetics questions. Deletion heterozygotes don't have sterility issues. Inversion heterozygotes, the sterility is proportional to the size of the inversion loop. So if you have a 10 map unit inversion, you have 10% sterility. As a result, we can see a range from 0 to 50% sterility depending on the size of the inversion loop. Translocation heterozygotes, on the other hand, are always 50% sterile. And the reason for that is because this is dependent on independent assortment. Independent assortment, 50% of the time, will segregate non-compatible chromosome structures. So whereas the sterility here is based on crossing over, the sterility here is based on independent assortment, and that's why it's always 50%. So when you're faced with a question on an exam or a problem set, you can use this chart as a way to help you identify what you're dealing with in a particular question.